everyone, I am Shwelidash welcoming you to the Medical and Pharmacy Crash Course Division in the Knowledge Club online. At previous episode, we learned about the action potential of nervous tissue. Today, I will discuss on the communication between neurons. By the end of this session, you will be able to explain the differences between the types of graded potentials. Stay tuned up to end because if you skip the lecture, you can miss valuable points in between lecture for your exam. So stay tuned up to end. I am going to start today's episode. Let's start. The electrical changes taking place within a neuron as described in the previous episode are similar to a light switch being turned on. A stimulus starts the depolarization, but the action potential runs on its own once a threshold has been reached. The question is now, what flips the light switch on? Temporary changes to the cell membrane voltage can result from neurons receiving information from the environment or from the action of one neuron on another. These special types of potentials influence a neuron and determine whether an action potential will occur or not. Many of these transient signals originate at the synapse. What is the graded potential? Local changes in the membrane potential are called graded potentials and are usually associated with the dendrites of a neuron. The amount of change in the membrane potential is determined by the size of the stimulus that causes it. In the example of testing the temperature of the shower, slightly warm water would only initiate a small change in a thermoreceptor, whereas hot water would cause a large amount of change in the membrane potential. Graded potentials can be of two sorts. Either they are depolarizing or hyperpolarizing for a membrane at the resting potential. A graded potential represented a change in that voltage either above -70 MV or below -70 MV. Depolarizing graded potentials are often the result of sodium ion or calcium ion entering the cell. Both of these ions have higher concentrations outside the cell than inside because they have a positive charge. They will move into the cell, causing it to become less negative relative to the outside. Hyperpolarizing graded potentials can be caused by potassium leaving the cell or clintering the cell. If a positive charge moves out of a cell, the cell becomes more negative. If a negative charge enters the cell, the same thing happens. Then what are the types of graded potentials? For the unipolar cells of sensory neurons, both those with free nerve endings and those within encapsulations, graded potentials develop in the dendrites that influence the generation of an action potential in the axon of the same cell. This is called a generator potential. For other sensory receptor cells such as taste cells or photoreceptors of the retina, graded potential in their membranes result in the release of neurotransmitters at synapses with sensory neurons. This is called a receptor potential. A postsynaptic potential PSP is the graded potential in the dendrites of a neuron that is receiving synapses from other cells. Postsynaptic potentials can be depolarizing or hyperpolarizing. Depolarization in a postsynaptic potential is called an excitatory postsynaptic potential EPSP because it causes the membrane potential to move towards threshold. Hyperpolarization in a postsynaptic potential is an inhibitory postsynaptic potential IPSP because it causes the membrane potential to move away from threshold. So what is the summation? All type of graded potential will result in small changes of either depolarization or hyperpolarization in the voltage of a membrane. These changes can lead to the neuron reaching threshold if the changes add together or summit. If the total change in voltage in the membrane is a positive 15 MV, meaning that the membrane depolarizes from minus 70 MV to minus 55 MV, then the graded potentials will result in the membrane reaching threshold. For receptor potentials, threshold is not a factor because the change in membrane potential for receptor cells directly causes neurotransmitter release. However, generator potential can initiate action potentials in the sensory neuron axon, and post-synaptic potentials can initiate an action potential in the axon of other neurons. Graded potentials summit at a specific location at the beginning of the axon to initiate the action potential, namely the initial segment. For sensory neurons which do not have a cell body between the dendrites and the axon, the initial segment is directly adjacent to the dendritic endings. For all other neurons, the axon hillock is essentially the initial segment of the axon, and it is where summation takes place. These locations have a high density of voltage-gated sodium channels that initiate the depolarization phase of the action potential. Summation can be spatial or temporal, meaning it can be the result of multiple graded potentials at different locations on the neuron, or all at the same place but separated in time. Spatial summation is related to associating the activity of multiple inputs to the neuron with each other. Temporal summation is the relationship of multiple action potentials from a single cell resulting in a significant change in the membrane potential. Spatial and temporal summation can act together as well. Revise it. I am damn sure you will require less time to remember all these structures. Write me at the comment box how much you enjoyed and time take to remember the nervous tissue anatomy physiology after reading to this lecture. Next episode we will discuss on the synapses and neurotransmitter. If you like this video, please like, share and comment freely. Don't forget to subscribe our channel. If you already subscribed, then thanks a lot. After subscribing our channel, please click on the bell button for getting the regular updates. I am coming with another video very shortly. Till then, goodbye.